Morning, everyone. My name is Christine. I'm a junior studying Africana Studies, and it's my honor to introduce Dr. Barbara Beltz. Dr. Barbara Beltz is an Allene Lummis Russell Professor in Neuroscience at Wellesley College, Wellesley, Massachusetts, where she serves as Director of the Neuroscience program from 1996 to 2016. She's also the lead consultant to the Sherman Fairchild Foundation for the undergraduate scientific grants programs and a member of the corporation at Woods Hole Oceanographic Institution. Dr. Beltz joined the faculty at Wellesley in 1987 after receiving a PhD from Princeton in 1979 and postdoc training in the Department of Neurobiology at Harvard Medical School. She has published over 90 peer-reviewed research articles as well as 2 books. Her work focuses on the relationship between the immune system and the nervous system, and specifically how the immune system is involved in the production of the new neurons in the adult brain. Please welcome Dr. Barbara Beltz.
Thank you very much. All right, so today what I want to talk to you about is this topic, world hunger and malnutrition. And what I'm a neuroscientist, and so what I want to think about with you and tell you a few stories about are studies that demonstrate that stress and undernutrition, lack of nutrition, have very long-lasting effects on the brain and how it's functioning, which I'll give you a little bit of a spoiler to start and tell you a bit of the punchline, and that is that the effects of malnutrition and of stress, which is obviously part of the same ball of wax, that I'm going to demonstrate for you that there are very long-lasting effects on the part of the nervous system that is important in learning and memory, and also the stress response system. And so individuals who have had long-term issues with conflict, with lack of nutrition, these individuals, just solving the nutrition problem is not going to overnight solve the problem, because they are going to potentially persist for many, many years, potentially the rest of their lives, with deficits that relate back to that stressful period. And so that's where neuroscience does come into some of these issues that are really global and that may influence in the long run how we solve some of these problems. So that's what I want to talk to you about today, beginning just by reviewing this report that I asked you to take a look at about world hunger and malnutrition. That was a multi-agency report. All right, so the talk will be in 5 parts that will follow through the next hour. What's the current status of hunger and malnutrition in the world? And by no means, this is not my expertise, but I think we all should have a certain level of understanding and appreciation for what these problems are, because it impacts our fellow citizens around the world. Secondly, a primer on brain structure and function. I'm very aware that a lot of you are not neuroscientists. Most of you aren't. And so I want to give you just the elements that you really need to know. And then to talk about the topic that's nearest and dearest to my heart, which is the fact that your brains are making new neurons all the time. Specifically in the area that is, I'm going to show you, is important for learning and memory. And so we'll talk about what's known as adult neurogenesis, that even after birth, the brains of humans as well as other organisms continue to make new neurons. Then the sort of crux of the problem, how do malnutrition and stress modify brain development? And this is, I'm using development loosely here. This does not mean necessarily prenatal, although my example will be one from prenatal. But really, brain development goes on through your whole life because you are adding new neurons. And so you should think of development of the brain being a continuum throughout your entire life. It's not downhill when you're born. You still make new cells, new neurons. And then finally, a wrap-up and discussion. So first of all, then, what's the current status of hunger and malnutrition? So this was from just September, a few months ago. A headline, Global Hunger Continues to Rise, New UN Report says. And so this is a huge disappointment. It's now at a 10-year high after having fallen for about a decade. And so we seem to be losing ground. And there are some reasons why that may be, that we may be able to understand this. Here is a global map showing where the hotspots are in terms of hunger, and with the warmer colors being the more extreme situations. And so you can see that there are a lot of areas in middle and southern Africa that have over 35% and over malnutrition. That's very extreme. Also Iraq and Afghanistan, and then other countries that are less so, but still certainly are experiencing extremes that would be unusual for us to understand here or to appreciate based on our own situation. We do hear about hunger and lots of children, even in the US, who are needy, who have this issue. But nevertheless, we're at the end of the spectrum where this is not outstanding compared to other countries in the world. And one thing that you may appreciate just from looking at this, which this report that you had to read made the point that many of the areas that experience the worst situation in terms of malnutrition and hunger are areas where there's conflict. And so this can be for many reasons. It may be that the lack of resources, the lack of food is part of the conflict. It's part of what exacerbates differences of opinion. When resources are scarce, animals and humans included tend to become more aggressive. And so there may be some of the roots of conflict in the hunger problem, and vice versa, that once the conflict gets going, this disrupts supply lines. It makes it harder to have food reach people. So distribution may be a problem. All right, so this shows you how really it is a global issue to a greater or lesser extent. All right, and so here's the actual graph that the headline a moment ago referred to. And so you can see that we've had a steady decline since about 2005 that reaches a nadir in about 2015, and then 2014, I guess, in terms of the actual numbers here. So percentages are in gray, and the actual numbers are in gold here. And then this is an estimate, but it has recently been confirmed, although the report was not updated. So you can take these numbers as being pretty reliable. And so you can see there's this pretty dramatic upswing in the last year or so, preceded by a small increase prior to that. So the concern is that we're losing ground instead of gaining ground. And there was a pact, a multi-agency pact that related to sustainable future that had a target of the year 2030, which I think you saw in the report I gave you, for solving these problems of hunger and malnutrition. And so this is taking us obviously in the wrong direction, and we've only got about 15 years, less than 15 years to go. Okay, so that's part of the challenge. Now, this is an interesting facet that it shows you how complex this problem is. This is just a correlation. Do you understand what the difference is between a correlation and causation? Who can tell me what their understanding of that is? If I say this is a correlation, what does that mean to you, and how is that different than causation? Anybody? Yeah.
A correlation means that there's an association between two trends.
Right.
Whereas a causation means that you've actually proved that one thing causes another. And generally it's easier to measure correlation, but it's very difficult to measure causation.
Exactly, exactly. So a lot of the graphs you look at, you should always think of this when you're looking at graphs. What is this telling me? Is it about correlation or causation? And certainly in terms of this kind of situation, it will always be correlation, because causation is very difficult to demonstrate. Even in science, when you can do a controlled experiment, it's still difficult to demonstrate causation. So this is correlation, but what it's showing you is that the number of undernourished people here, and these histograms, is much higher in countries affected by conflict. We can't know whether the hunger is causing the conflict or vice versa, or some of both, which is probably the answer. And then, so this is the countries that are affected by conflict and not affected by conflict. You have prevalence of undernourishment. They call it weighted here, which means it's been normalized. So this is the y-axis you'd use. It's a percentage. This is the mean, and this is the unweighted here, looking at the actual numbers, okay, the means. So that gives you a little clue about an association at least. And here, the scary part of that report for me was looking not something, it is something we know from reading the newspapers, but to see it in a quantitative way, this tells you an increase in the number of conflicts since 2010. It also suggests, because this is a non-state violence, and if you look at the definitions of this in the report, what this means is that these are warring factions within countries that is on the increase rather than wars between countries in general. That this has superseded the wars between countries, which is in the gray here, okay? So, but both are on the rise, okay? So that, if we then take that and associate that then with incidents of hunger, then we can assume that with the increase in conflict, we will be also seeing some kind of increase, probably in malnutrition and hunger in those people. They're displaced. Usually those displacements are over a very long period of time. So we're going to see probably that coming out in some of the hunger reports in the next few years. All right. And so that's just the high points of that report that you have that you can refer back to. So I first then want to give you a little bit of a primer on brain structure and function. Just 3 slides. And what we're going to be focusing on today is the hippocampus, which is this C-shaped area in the brain. It's bilateral, so it's on both sides of your brain. It's in blue. It plays an important role in learning and memory. If you have hippocampal injuries, you can't form new memories. All right, so that's been something that's been known now for decades. It's done, and that's due to damage in humans that has confirmed what we also know from preclinical studies in mice and rats. Preclinical meaning some of the models that we use to model ourselves, okay? So there's a huge amount of evidence that the hippocampus is critical in the day-to-day sort of remembering of facts and learning along the way, okay? So that's what you need to know about the hippocampus at the very basic. Now, the second point that I want to make is about the hypothalamic pituitary adrenal axis, also known as HPA axis. And this is an axis that runs from the hypothalamus, as the name suggests. Stress affects the hypothalamus, which through a couple of intermediary kinds of hormones that are released from the hypothalamus and the pituitary affects the adrenal gland. The adrenal gland produces, among other hormones, cortisol. And cortisol is a steroid. It's a glucocorticosteroid that is involved in the stress response. So here it says physiological changes supporting fight or flight responses. So cortisol itself is released then in response to stress and also in response to low sugar levels in the blood. So if you're undernourished, that's automatically interpreted by the brain as stress. All right, so undernourished people will hyperactivate this system and release more cortisol, is the point. Cortisol releases blood glucose, is one of its many functions. And so it will raise glucose levels in the blood, which if you're walking across the street and you see a big truck coming at you and you have that, that fight or flight sort of response that I got to get out of the way here, cortisol is going to be released very rapidly, raises sugar levels, allows you to run faster than you ever knew you could run. So the blood sugar levels are a critical part of the response. The second response is that it will reduce immune activation. The immune system will be suppressed. And that's important to think about. It was inferred in the second article that I just asked you to take a look at, but not to read necessarily from word to word. Because when you have suppressed the immune system, individuals with that kind of a situation are going to be more prone to disease. They aren't going to be able to respond as a normal person would without a compromised immune system. They won't be able to respond to disease threats. Okay, so those are two of the many effects of cortisol. It's obviously more complex than this, but that's all you need to know for today about that. And then here's the third piece, and that is that this is the HPA axis that releases the cortisol. The amygdala is a part of the brain that's involved in emotional processing. And so if you're very angry or very upset about something, the amygdala is part of that response. It's not the only thing the amygdala does, but it's certainly an important part of the pathway. The amygdala will excite this HPA axis or cause it to be more active, which means it would release more cortisol. When cortisol is released, it acts on the hippocampus, the learning and memory area, and so will make the hippocampus more active. And the hippocampus, in turn, suppresses the HPA axis. And as far as we know at the moment, these are the two primary players in this regulation of the HPA axis, the sort of push-pull, that we've got the amygdala driving the HPA axis and releasing cortisol, and on the other side we've got the hippocampus suppressing to some degree the HP axis. All right, and so how that balance goes will be really critical in how much cortisol you release and what kind of stress response you have. If you have too much input from the amygdala, you'll have an overactive HPA that will cause you to be a hyper-vigilant kind of response physiologically. Question?
So does that hypervigilance influences the hippocampus? Like, what impact does that have on memory?
Okay, we'll get to some of that, but I'll answer this now. So while cortisol does drive the hippocampus activity, in fact, cortisol also reduces the production of new neurons in the hippocampus. The hippocampus is one of the primary places where new neurons are being added in your brain. And so cortisol reduces those numbers, which means you're going to have fewer neurons functioning in the hippocampus, which will ultimately reduce the drive, the negative drive on HPA. So if you have less equipment here, basically, then there'll be less negative drive, which will tilt the balance towards the excitation from the amygdala. Okay, and so that is part of the story. In fact, that's part of my punchline at the end. Okay, but you can be thinking about it, and if you're not quite there yet, we'll be coming back to that at the very end. Okay, great question. Okay, so 3 important points. The hippocampus is the brain structure critical for learning and for establishing new memories. Stress causes the release of cortisol and the stress response, which is this production of glucose and suppression of the immune system. And then the hippocampus is also involved in regulation of this stress response. Okay, so those 3 things are sort of the basic foundations for where we're headed. All right, so moving on then, new neurons and old brains, adult neurogenesis. This is what I've spent my career over the last 20 or so years working on. I actually work in an invertebrate model. I work on crayfish and lobsters and crabs, those sorts of animals. They also make new neurons throughout life. And what we're doing in my lab is to study basic mechanisms to try to understand how the regulation of these processes is controlled and where the stem cells are coming from that are producing the new neurons. And so there probably are some far-reaching implications I could bring in from my own work, but I'm going to stick with other literature that is closer to the human condition today. Okay, so definition of adult neurogenesis, that stem cells in the brain divide and differentiate into neurons. Okay, they'll divide many times, as you'll see, but then the final descendants will differentiate into neurons. And as I mentioned, the human brain, neurons are added to the hippocampus. In fact, this is the area of the brain where we integrate neurons at the fastest rate. And there's quite a bit known now about what they add to memory and learning function. Just to give you a little glimpse of that, what's thought is that the new neurons are adding an ability to distinguish memories in time rather than clumping a lot of things together. So it gives you a more refined ability to retain memories and to form those memories. An example from the human experience might be, I think you've probably all heard of individuals who have had chemotherapy and saying they've got chemo brain. And at first people weren't thinking this was more of a psychological issue, which is still brain, but psychological. It's now quite well known that chemotherapies, we were using these long before we were aware of adult neurogenesis. Turns out chemotherapies are stopping the division of all cells in your body. It's trying to slow down divisions in your tumor. And unless you're using an immune-targeted therapy that targets just to the tumor, a standard chemotherapy will stop cell division of your blood cells, which is why often after chemotherapy you have to boost the blood products in order to get the blood back to where you're feeling energetic again. But it will stop the cell divisions of all types of cells, including the neurons in your brain. And so what's believed now is that chemo brain, this sort of fuzzy feeling about the way you're thinking and that you're not as sharp as you were before the chemotherapy, that it's real. And it's about losing part of this ability to refine your memories, to really think as clearly as you did before. It's probably related to hippocampal function and the fact that during the chemotherapy you are slowing down the production of new cells that you need for those functions. Yeah?
Why would the targeted chemotherapy not be used under temporal?
Now, these days, a lot of these are targeted immunotherapies. And so we're getting around that, which is a huge, huge advance. Technological advance, being able to do this. And so the whole point is with the immune therapies that target the specific tumors, you're not getting the side effects because the therapy only goes to the tumor rather than being a systemic therapy that is going all over the body and affecting your blood cells, affecting your brain, that it's a much more targeted activation. Okay? All right, good. So there are also a lot of other areas that incorporate new brain cells during adult life, adult being postnatal, but the hippocampus by far has the most robust neurogenesis, okay, most robust. Yeah, so that's what's important for today. So here is, again, a picture of the hippocampus now from the side rather than the view we saw before, the C-shaped structure in the middle of the brain. It's organized into various layers, and what's shown here is the progression of what happens when new neurons are being produced. You start with a stem cell, these green triangular cells here. They do have some processes. Neurons are not the only cells that grow arms and legs sometimes. And so the stem cells do have processes. They're sometimes called type 1 cells. They will divide. Every one of these circles here is another mitotic division, and I'll remind you about mitosis in just a second, what that does. But you're getting basically a doubling of cells every time you have a division. And then after about 3 divisions, then the progenitors will begin to, these descendants will begin to differentiate into neurons. And once they become a neuron, they can't back up. All right, they become a neuron, they're a neuron for life. And so you go through many intermediate cell types before you get to the actual neuron. And this is the tissue itself. The subgranular zone is where this is going on. The granular cell layer is where the cells are integrated. So these will be what are known as granule cells in the hippocampus. A little sidelight here that sort of is a prelude to where we're going. Has some ups and down arrows here, which lists some of the factors that affect different cell types in this maturation process. And so one thing you can see here is that antidepressants will increase the rate of production of cells. Running, exercise increases the rate of production. Decreases come with drugs of abuse and stress. Stress decreases the production of new cells in the hippocampus. And so you can look at that later. I believe that will, and stress functions at a variety of levels through this process. All right, and then learning also increases the production of the final neurons. All right, yeah?
I'm just curious how long this process takes and like, it's a long process.
Okay, it takes about 2 weeks.
Okay.
is what our understanding is from preclinical sorts of studies. So about 2 weeks to go from stem cell to neuron, and then it takes several more weeks to be fully differentiated and to be fully functional. But these young neurons are actually integrated into circuits at about 2 weeks of time before they're fully mature. And we think that the function they're contributing actually is related to that maturation process. So it's a little too much to go into today and without any graphics to help. But they're incredibly interesting cells, and their contribution is becoming more and more appreciated all the time. Another question somewhere here.
So here when we're talking about stress, is it like high stress in terms of what you mean in the larger part of the presentation, or is that also?
It can also be our day-to-day stress. As you probably read in just the newspapers, our immune system and the stress system in general is calibrated. It was originally designed to respond to threats like being chased by a lion, for occasional threats that are major, that allows us to respond with an instant burst of energy and getting out of the way. But the use of this system, this HPA axis, and the immune system has evolved over decades and centuries to the point where now many of our lives are stressful, not in the way that armed conflict is, but it's still a type of stress that affects the HPA axis. We're getting to the point where we have stressful types of lives day to day rather than the occasional that we believe the system was designed for. And so there's quite a bit of talk about that in the lay literature related to how this constant exposure to increased levels of cortisol affects us all the time. So I think even for those of us here, we're in a relatively incredible environment with good things happening, challenges that we're learning. And I always sort of talk about good stress because sometimes when I get to the end of a day at Wellesley, I feel like I just finished the marathon. And I feel like that was stressful, but it was good stress. And I think there may be a distinction there if you're enjoying what you're doing, even though you're high energy and it is stressful as well. There may be some differences, and I don't think the literature or the research is to the point where we can distinguish some of those things. But the negative stress that you certainly feel, if you had to take a final exam every single day, that would certainly fit. And for the CEO that is under fire all the time, it counts. It counts. That is stress. All right. The form of stress that we're talking about during armed conflict and malnutrition is an extreme form where you'll see even more outcome. But certainly your day-to-day stress does count. So the running is good. It relieves stress. It works against the negative stress factors. And so it's a good thing to watch for all of us. So mitosis. You all would remember this regardless of what you're doing at Wellesley, back to probably about 6th, 7th grade. And this is all we're talking about with adult neurogenesis is the fact that you have this cycle that cells that are dividing go through. They go through a growth phase, then a DNA synthesis phase, the S phase, another growth phase, and then they will go through mitosis, divide. And in the case of a stem cell, one of those stem cells will stay in the cycle, as this one is here. The other one usually leaves the cycle and will do something else. And so for the situation we're talking about here with neurons, this cell would go through a few more divisions potentially, but then go into a part of the cycle known as G0. They lose their ability to divide when they begin to differentiate. So this would be the presumptive neuron after a few more divisions that would then become a neuron and never go backwards. So once usually a cell is differentiated, it's rare populations of cells that can actually back up and go back into mitosis, okay? And that's kind of important in some of the techniques that we use that I'll be mentioning. And so when we're studying adult neurogenesis, we want to be able to quantify how many new cells are born under different kinds of variables. And so what we primarily do is to use a molecule known as bromodeoxyuridine, or BRDU, and this is incorporated during this DNA synthesis, or S phase, to label the newborn neurons. And you may remember that DNA is made up of 4 nucleosides: adenine, thymidine, cytosine, and guanine. Right? Does that sort of ring a bell? Okay, back to 7th grade or something like that. Yeah, maybe high school, I guess, for that. And so bromodeoxyuridine is an analog of thymidine. And so if you were to inject BRDU into an animal, it would be in higher levels than the endogenous thymidine, and so it would be taken up in proportion to the relative levels in the body. BRDU has also been used in humans to track the progress of tumors and to test whether certain chemotherapies in clinical trials are actually working well. You can inject BRDU for that kind of a purpose. It's not a molecule you'd voluntarily take on, but if you're in a clinical trial, then you can inject that into a human, and that's how the first human studies of adult neurogenesis were done, was to then have people that had been involved in clinical trials donate their brains for science, so that when they died, people could look for the incorporation of BRDU in the brain itself. And since there are only a few areas where we're aware of new neurons being born, and because once they become a neuron, they may last you the rest of your life, once that BRDU is into a cell, it stays there. So you've really got a historical record in the BRDU labeling. Okay? And so the cells are being born in the brain by mitosis, pretty simple process. And we can use this part of the process to label the cells that are going through that and producing the new neurons. And the new neurons will contain that BRDU. Even when they divide, they will divide the BRDU as well, but all of the descendants will, in a lineage, will contain the BRDU. Okay, so here's a DNA, and this is as technical as we'll get, and then we're going to move to other data that's more accessible. But I want you to know how we know what we know. This is not magic. Okay. All right. So here's a strand of DNA, and when you're replicating the DNA in the process of S phase, leading into mitosis, the parent strand of DNA unwinds, and here are the new strands being made as we go, as we unwind. Now, those 4 nucleosides pair in certain ways. Adenine and thymidine always pair with each other. And so what's going to happen with the new daughter strands that are being born is that BRDU will replace the thymidine at many of those sites in the new strands. Okay? So it'll pair always across from an adenine. And so BRDU is where the thymidine was before. So in these sorts of linkages, you'd have BRDU. Then we use antibodies. This is an antibody-based method. Antibodies generated against BRDU, and then usually a second layer of antibodies generated against the first group, and these have a visual marker. And so this visual marker, the green dots here, those then indicate all of the sites where BRDU is present. Okay, so when we see this fluorescent green label potentially, you can use various kinds of labels. When you see that, that tells you that that cell just went through S phase, okay, or has gone through S phase since the time that you injected the BRDU. So you could inject the BRDU and wait 6 months and look to see where those cells are. Okay, the BRDU is gradually taken up in the system by the metabolism, by other tissues. Any tissues that are dividing will take it up. It's not special to the nervous system. So your liver is dividing. Those cells are dividing all the time. That will take it up. All right, so over the course of hours or days, the BRDU levels drop again. And so what you'll see later on, whenever the animal is sacrificed, or if it's a human study and the individual dies, and you study their brain, you'll see what cells were born in that little window at the time you injected BRDU. All right, so it's a really powerful technique. All right, so Here's an example. This is BRDU labeling, allows visualization of the new neurons in the hippocampus. Here's a little piece of that C-shaped structure that I showed you before. Here's with a label that you can use under a traditional microscope. It's an enzymatic label rather than a fluorescent label. So all of the dark blobs here are cells that were labeled during S phase, after an injection. This would have been in a mouse. Okay, and here's another example with even more cells, and the fluorescent label of a different section from the same. Here you're looking for the orange cells. I don't know what the lights as bright as they are, as you can see this very well, but you can probably see some orange cells peeking out through this. Okay, so that's what the BRD labeling looks like. And as I actually was going to ask you which you thought was more, but it's kind of obvious. All right, so this is actually part of a study on the hippocampus, and it turns out these were control mice. These were running mice that had wheels in their cages. And they are producing, depending on the protocol, they can produce 2 or 3 times as many cells because of that exercise and the running. Okay? So this is the kind of thing where you would go through and you'd count every cell here, or the orange cells here. The green cells are all the other neurons that are there, but if they aren't orange, this is done with a confocal microscope, the observation, and you can separate out or merge different kinds of antibody labeling. And so you're getting the orange because you're merging the BRDU label with the green label that identifies the cells as neurons. Okay? Is everybody sort of staying with me? This is as technical as it's going to be. All right, yeah.
I have a question with regards to the running and the exercise.
Yeah.
So is it that because of the energy used in the running, is it that the energy used in the running is like, is that the energy that causes the formation of the new neurons, for example? Okay, my question is, for example, I walk a lot, right, as opposed to running.
That's good too.
So I wonder whether
any kind of exercise like that. I mean, you could sit in your chair and move your ankles around, and it would potentially help. So what's happening when you exercise, any kind of exercise? You're going to change some of the physiological and hormonal balances in your body if you do this routinely. I'll give you just one example. It's not this simple. But when you exercise to the point where you're thinking, this is exercise, even the walking a mile or 2, whatever, serotonin levels go up. Now, serotonin, you probably have all heard that word. It's associated with clinical depression. Reductions in serotonin are. Serotonin actually is like a magic bullet with adult neurogenesis. Serotonin raises levels of adult neurogenesis. It's basically a good hormone. It does all sorts of amazing things throughout the brain and also in the body, peripherally. So one of the things you're doing is to change the levels of certain kinds of hormones. If you really are doing lots of exercise, of course, you've heard of runner's high. That's associated with a particular peptide that's produced in the nervous system that gives you that feeling. And serotonin levels also rise. So really, any kind of exercise. The running with mice is the best way to do a controlled experiment because they love to run. In fact, there was a recent study, this is off target, but a recent study because I think especially, well, all of us, you wonder sometimes when we put animals through these variables, how realistic this is to what really goes on. And some people got very curious because wheel running with mice is such a prominent technique used to demonstrate exercise and to look at the outcomes with mice. Someone put wheels in an area of woodsy area just within the last year or so. It was published to see whether wild mice actually like to run on wheels. They do. They actually will find those wheels and they run on the wheels in the wild. Isn't that amazing? Yeah, so this is not so unusual. Yeah. Now, there was one more hand. Yeah.
So just out of curiosity, for lacrosse, I have a run test.
Yeah.
And while I can understand the physical action of running will increase serotonin levels, is there a similar, I guess, for me, sometimes it feels like a final exam because on that day, I need to pass.
It's also stressful. It's a balance, so it's going to be complex. And so if you're in the middle of the very heated, challenging game against an opponent, you're going to have stress hormone, you're going to have cortisol being released. You'll also probably have serotonin levels rising. And what you're going to see in terms of the physiological outcomes is going to be a balance between the two. You'll see all of that stuff adds together. It's like a really amazing spreadsheet of hormones that are changing up and down, and the outcome is a blend of the bunch of them. So you can have all these things acting in parallel, and you'll have an outcome that is a blend. Yeah. But overall, exercise is great. We should all be doing it. Okay, was there another question? Okay. All right. So, so you can see here that the labeling is easy to see. You can count these cells. We can quantify how many are born under whatever circumstances we want to look at. All right. So that's the background about adult neurogenesis. So now to the crux of the story: how do malnutrition and stress modify brain development? And let me just remind you that malnutrition, because you're going to have low sugar levels, is actually a form of stress in terms of the physiology of your body, because that is going to be interpreted by the HPA axis as a stressor. Low sugar levels activate the HPA axis and will release cortisol, right? Which will activate the hippocampus, which should suppress then and balance. But so this is what I want to talk about next. And what I'm going to discuss actually is a specific study related to stress in rats, but this is sort of a prelude. This was one of the original studies looking at regulation of adult neurogenesis in the hippocampus, where they found that if they put mice into an enriched environment, and that was a large cage with lots of friends, toys, tubes to run through, challenging things to do, running wheels, that animals in that environment made many more neurons than mice that were in standard cages with no running wheel, by themselves, sometimes solitary kind of living. And so this gave everyone the idea, aha, enriched environment. So what is it about the enriched environment that's making the difference? And so we're still trying to tease that apart. But the same is true actually in crayfish. We can put them in what a crayfish would consider an enriched environment, or at least we think that that's what they would think was enriched. And you also see this difference. So enriched environment involves social interaction potentially, space to move around, exercise, challenging things to do, etc. And so the field has been trying to work out what these positives and the negatives in the case of the controls or the deprived animals, what that really means. And so this was sort of in my interpretation, one of the sort of motivations for the studies that came later. And one of those was this one that I want to go through actually the graphs with you, because I think the message that we'll see coming from this is really important to the discussion of what's happening in the world with malnutrition and conflict. So the title, Prenatal Stress Produces Learning Deficits Associated with an Inhibition of Neurogenesis in the Hippocampus. All right, so prenatal stress, learning deficits associated with it. So I've told you that adult neurogenesis seems to be, well, in the hippocampus, is part of the machinery that will promote learning and memory, right? So if you reduce levels of neurogenesis, as with chemobrain, then you will affect the ability to learn. And so this is one of the studies that actually demonstrated this. Now, this is the sort of technique leading up to it. And the situation was that pregnant female rats were exposed to stressful environment for the last week of the pregnancy. And it was the pups that were then assessed. So the offspring is the data you're going to see. So it's pretty amazing because the stress was affecting the HPA axis of the mother. What they did was to restrain the rat. They don't like to be held down. They were put in a close sort of environment where they couldn't move a lot. And then bright lights. They don't like light either. And so this isn't a horrible, stressful situation, but it's not, but it is stressful for them, all right? But it's sort of intermediate, maybe equivalent even to some of our stressful days, okay? That you don't like bright light, you don't like being enclosed, but sometimes you're in those sorts of situations. So I would say mild stress. All right. And then there were control pregnant female rats that were left undisturbed, so just standard procedure. And then the offspring were reared by the mothers until they were weaned, which is around day 21. And then the pups were injected with BRDU, sacrificed at various times later. As you'll see, it was a longitudinal study, so different groups were examined at different times. And they quantified the numbers of labeled neurons in the hippocampus. And so remember this pathway, stress, ultimately altering levels of cortisol. That's what we are talking about here. And one way we know that this was stressful for the rats was looking at the pups. This is the offspring now, so not the mother. And so this would be cortisol from the mother influencing the development of the pups. At 28-day group, 3 months and 10 months. So 3 different groups of offspring that were assessed at different times. This is actually the weight of the adrenal gland that is producing cortisol. And it's known from for a long time, various studies, that when in fact the adrenal gland is hyperactivated and overproducing cortisol over a long period, it actually gets heavier. It's a bigger gland. And so here you can see that 28 days after birth, this would be the first group, the earliest one assessed. The prenatal stress group had a slightly higher weight. And then look at what happens. That's not that different, although it's different. But you get to 3 months, and it's even more extreme, the difference. And this is the pups, who long since left the mother's womb. And they're still, even though now they're in a very normal environment for a rat and experiencing normal conditions, the adrenal gland still has some kind of history that is being played out here. And so it's continuing to release, presumably, more cortisol. And then at 10 months, it's a little bit back towards this level, but it's still higher than it was at 28 days, all right? So this is telling us, aha, these pups that long since left the exposure to the mother's cortisol are having some kind of a lasting effect in terms of how the adrenal is working. Okay? And this is just 1 week of exposure at the end of pregnancy. The rats, their pregnancy time is about between 4 and 5 weeks, okay? And so it was just the last week. So they'd gone through most of what we would think of as the early neural development, brain development, and this is just at the end. And it's not extreme stress in the sense of, you know, any sudden damage or whatever. It's not what a scientist would consider extreme, kind of mild, intermediate stress, okay? And this is the pups. I just have always found this remarkable, this whole set of data, which is why I wanted to share it with you. All right, and here's the hippocampus. You can see part of the C here. This is in the control pups, lots of new neurons. Here's the stressed pups. It's really prenatal stress, but they've got very few cells. And you can just eyeball this. You don't even have to count. You can see the difference. This is dramatic in terms of science. Quite often, you have to count every single cell to know whether you see a difference, and you have to have a huge sample size in order to have with any confidence what the result is. Yeah?
So if now the stress pups have kids, will they have normal levels?
That isn't known. That isn't known. But let's come back to that. Ask that again at the end, because I think it's a good discussion point. If we think that these stress pups have an hyperactive HPA axis, and then the pups will be sensing that, I would predict that there will be some effect. Maybe it's going to be moderated to some degree, but let's come back and think about that some more after you've seen the rest of the data. All right, now here's some of the data behind the conclusions I've already shared with you at some level. Prenatal stress decreased cell proliferation as compared to controls. The prenatal stress pups are the black histograms here. They're on a different scale at 28 days than at the later groups. And what you can see is that the 1 star indicates that this is statistically different. The prenatal stress group, the actual numbers of BRD labeled cells. And then when you get to 3 months, we've got 2 asterisks, and 10 months, 2 asterisks. And that means that it is even more highly statistically different. So there's even a bigger variance between the control and the experimental animals. Okay? So that suggests that the situation in terms of hippocampal neurogenesis is actually becoming worse even at 3 months after birth than it was at 28 days. Now, we don't have earlier data because they were living with the mom, lactating, and not weaned until 21 days. And so they've had basically a month, 28 days, of very normal maternal care. All right, and those mothers, according to the report, behaviorally seemed to be fine. So it was just this period of stress that then ended. Okay, and then they took care of their pups as any normal rat mom would. Okay, so you're seeing big differences in the BRD cell numbers. We don't need to go into detail. These y-axes are very different at the later times, and that's because there's also a process of neuron death going on. As cells are born, some will die. We sometimes talk about it as Darwinian selection at the level of cells, that the ones that are functioning the best, that make the best synapses, that are most active, are the ones that remain, and some of the others will be weeded out. And it's pretty dramatic. It can be half or more of the cells that are born. So that's why those are different scales. All right, so over a period of time then, you'd expect that if you're making fewer and fewer hippocampal cells all the time, your hippocampus does normally grow throughout life. If you're making fewer cells all the time, you should see that in the overall total numbers in the hippocampus. And so this is the total number of granule cells, which are what those new neurons will become, as I mentioned before. In the dentate gyrus is just this part of the hippocampus that's incorporating the new neurons. And so what you can see at 28 days, the hippocampus of the stressed pups is almost identical in terms of total cell numbers to the controls, black versus white bars. But as you get to 3 months and 10 months and 22 months, the difference in the size of these becomes greater and greater, even though their postnatal life was identical to the control animals. Okay? So it's this long-term process that is the concern that I want to present to you. All right, and so this is about a 30% decrease if you look at the actual numbers in their data. Okay, that's huge. 30% less equipment to work with in the area that's most important in learning and memory. Okay? All right, now, so an obvious question would be, are there behavioral and learning differences in the two groups, right? And so often the way this is tested is with the Morris-Water maze, which if you looked at the Neuroscience paper, they were referring to this, I apologize, as I didn't pick this up when I read it the first time before I put it on your list. MWM, they never defined it. This is terrible, and that shouldn't be that way. But it's the Morris-Water maze, and that's what you see pictured here. This is one that we had in the Science Center for someone's research. And so it's this tank here, which has a submerged platform in it. Okay, now mice and rats don't like to swim. And so it's kind of it's not a fun thing, but they do it. And so there's a submerged platform that they can stand on, but they have to find it. And usually you put milk in the water or something that'll make the water cloudy so they can't actually visually see it. So the first time out, they have to just swim around until they actually fall upon it, or if they don't ever reach it, then the experimenter usually after a minute or two will just take the animal by the tail and put them on the platform to help them understand where it is. And this is a spatial learning task because there are icons, I guess, on the wall, certain features that the animals will recognize, and also even in the tank, so that they do have a spatial orientation they can establish relative to where they are and where that platform is. Okay, so let's say the platform is right under this drain, or whatever that thing is, then they would say, it's right by the drain, and then if they learn that, they can go back there and then they don't have to swim as much. Okay? So that's sort of the point. All right, so here's what actually happens. And then before the learning, the first time in, a mouse or a rat will swim all over the place. It usually can take them a minute or two to find it, the platform, and to pull themselves out of the water. And quite often the researcher does have to place the animal on the platform that first time. They usually set what the limit is. After 1 minute, we're going to put this mouse on the platform, so then they know what the game is, okay? But after learning, which is usually about 10 trials that people run, then you put them in the water and they go right for the platform. Okay? And so what you measure in terms of the learning here is the latency. How long does it take them to get to the platform? So if it took them a minute the first time and 10 or 15 seconds the second time to get to the platform, that would be your measure of whether they have learned. Okay? Now somewhere here, there's a fun video that I didn't think we'd probably have time for, but you can watch trials online of an animal, a mouse in the first trial, going all over the place swimming, and the experimenter lets it swim for a couple minutes and then finally puts it on the platform, and then seeing that same mouse after 10 trials, and boom, right to the platform. All right, so here's the result. And so here's the latency. How many seconds? So a good score is shorter, fewer seconds. A shorter latency. And so the stressed pups, this is the mean up here, the latency for all of the trials for 5 days. And so you can see that the stressed pups are very different in terms of their ability to do this task. And then granted, it's one task, but there have been a lot of other types of variables that have been applied, learning methods that have been used. This is kind of the gold standard, but a lot of other types of tests support these results. And so what you can see here is that at every time point during this 5-day training period for this group of pups, then the control group is performing better. And overall is this mean data that's shown here. Okay? And this is highly statistically significant with 2 asterisks. So the pups that were stressed in utero for 1 week, even months later, are having trouble learning this task. They do learn it, but it always takes them longer within the time frame of the experiment as it was run. Okay? We don't know if you came back to these same pups a few months later, if it would have resolved, but given the fact that the hippocampus was basically going downhill, wasn't getting better in terms of the numbers at 10 months, wasn't that the outlier group? It's unlikely that this was improving, but we don't know that from this particular data set. All right, and so then one more graph, the last, from the same paper. And this is BRDU cell number, so labeling how many cells are being generated in the hippocampus. And in this case, what they were doing was training animals in the Morris water maze and then looking to see how many cells they had after the training period. And because the animals that are in the Morris water maze had been handled by the experimenters, they thought the best control was not an animal that was just sitting in its cage, but one that had been manipulated. All right, so they had a manipulated control that was handled sort of in the same way that the animals that were in the Morris water maze, and then the ones that went through the water maze. And what you can see here is, which is fascinating, and this has been repeated many times with different types of animals and in different situations, that the control group these are then also exposed to the water maze, but they were manipulated in advance. All right, those actually, as a result of being in the water maze, actually are producing more cells. And this has been found in many studies that if you put an animal in a learning situation, a challenge that they have to figure out, that they're making more neurons. Probably part of that original enrichment that I talked about that I just showed you the title for the original paper in '97, was the challenge of some of the toys and games that were in that housing. All right, so they learned, but the stressed pups in the Morris water maze, those, in fact, have somewhat fewer. These are statistically different than controls, but not from each other. So these, in fact, don't see an increase in the generation of new neurons after that learning. All right, so not only are they learning more slowly, they're not getting that boost from learning. I mean, I think this, in my mind, sort of relates to when you get into one of those zones when you're studying and, you know, things start to click and you can really hold that feeling for a while and you feel like you're working at a sharper level, that may be due to some of this over a long period. You know, if you're writing a senior thesis and for a couple of months you're just so, you know, involved in that, that it may be that part of what is keeping that energy going intellectually is this generation of new neurons. These pups that were stressed prenatally don't experience that, apparently. All right? They are producing some neurons, but at a much lower level, and they don't get that boost as a result of the challenge. Okay? So what that says is that learning promotes neurogenesis, but only in the non-stressed pups. All right? So when we think, therefore, I mean, we think about Head Start programs for human children and ways of enriching the lives of individuals that have had stresses, have had difficulties in their lives, and trying to put them in enriched learning environments. This can certainly help, and we know it does help in the United States. We have lots of programs that do this, but it may be that the physiology is fighting back to some degree. That it's maybe not just a simple, okay, if we enrich this, that they're going to then have their full potential. They may still not reach their full potential because the brain is not working in the same way because of this HPA axis. Yeah.
Yeah, so that's actually what I was wondering about, because they've done like a lot of testing that shows that physical stunting is related to a decrease in lifetime learning and also a very significant decrease in lifetime earning potential, which obviously has serious implications
this
for
is exactly
economies.
What you're looking at.
Yeah,
Yep.
So I'm wondering if there's, and then if also the brain can't learn as well or doesn't gain from increased enrichment, is there any way to recover any of that lost potential?
I think we're still learning that. These are all preclinical experiments. Certainly, you're helping with all the enrichment programs that happen, and bringing humans into a better environment is going to help. But I think the point is that it's going to be a long process. It's not something about, oh, let's solve the food problem in country X, and then 5 years from now, it's going to be a totally different situation. I think these people are going to have to bootstrap themselves probably through at least a generation. Mm-hmm. So it's a long-term support that we're talking about here in terms of the neuroscience. Yeah.
Follow up. So Ida asked the question about like the offspring of people who've been stressed, and I'm wondering if there's a way, if we can talk, if any studies have been done on that at all, because that obviously has implications like for recovering that like generation that might have been stunted.
To my knowledge, not. I haven't done an exhaustive hunt. It would be a wonderful thing to do. I would expect that the effect hopefully would be somewhat reduced, but I would also predict there'd be remnants. You'd still see some of it, just because of how resistant those pups are to modification. Yeah, so I think the answer to this is not clear other than long-term support, both in terms of food and suppressing conflict and all the other things that are going on. Yeah, go ahead, yeah.
The question I had was that this like study is focused very much on that 1 week, and I was wondering if there's been other studies that have looked at like, oh, well, what if it's like right in the beginning? What if the mother was stressed before she got pregnant and then was moved, or something like that?
Yeah, I think anything that exposes those pups to cortisol. And there have been a lot of postnatal experiments that have been done stressing the pups themselves. I mean, our ability to do these kinds of experiments is somewhat limited. I think they're prevented entirely from doing this in this kind of experiment in monkeys or in higher evolutionarily animals that are more closely related to us. And this is rightfully so, because all of these organisms deserve the respect. And so the government in the United States carefully controls what sorts of experiments can be done. But yes, young mice have been exposed to stressful experiences, and you see the same kinds of outcomes. And presumably, because they will have an increased cortisol production themselves, if they were then to become pregnant, their offspring would experience that, maybe at a lower level. It may be that when the pregnant mothers first experience that stress, they may have had a big surge of cortisol. And where if you've got just a steady state lower level, that may have slightly different effects, I suspect. So I don't have the answer to that, and I don't think that study has been done. But there have been lots of other studies that replicate this in different ways that all point in the same direction. Stress is really bad for adult neurogenesis, really bad for mechanisms of learning and memory. Okay, so that is consistent over many, many studies. All right, so just wrap up in discussion, a couple of slides to remind you of this. So I think what's going on here, the interpretation would be that we're getting this positive drive due to low glucose levels in undernourished populations or due to stress in areas of conflict. That's increasing the amount of cortisol, which is driving the hippocampus, because there are receptors for cortisol in the hippocampus that will activate the hippocampal network. And so electrical activity will increase as a result of cortisol, but the machinery here is reduced because you don't have all those new neurons. You've got less equipment, basically, less neural equipment. And so you're getting less suppression of the HPA axis, which means that you're tilting the balance towards hyperactivation through the amygdala. And that this will be a long-term thing. The only way to solve that would be to increase neurogenesis in the hippocampus. Now, we could probably do that by using antidepressants, because that will increase serotonin levels. At least many of those will. So you could do this chemically with pharmaceuticals. That might help, but imagining that we're working with whole populations to do this and to have compliance would be difficult. But in terms of fixes, in an ideal way, I can think of a few things that would help, but they're not realistic. Okay. All right, and then this is from the article the neuroscience article that I put in your packet, but that is quite technical in some ways. But starting with early life adversity, whether that's malnutrition, stress, infection, because remember the immune system has been suppressed, so it may cause additional health problems. And then what you get is this neuroendocrine stress profile, which is the cortisol balance. Nutritional profiles, immunological profiles, all of these things are affected. And this then will alter the hippocampal structure. And plasticity means how well it's able to respond to change, which normally the hippocampus should be able to regulate up, down, do all sorts of amazing things. It has reduced plasticity, and this leads to cognitive impairments in adulthood. And so going back to this map then, when we think of some of these countries that have this 35% and greater levels of undernourishment and malnutrition, this is a huge problem, not only from getting these people to a place where they're comfortable physically with their food access, but also the fact that they may be fighting to some degree a difficult battle based on what we know about the HPA axis and the fact that insults to that axis early on are not going to go away overnight just because nutrition is improved. All right, so that's really the big take home. And I want to come back to this, which my colleague yesterday showed you, and I was fascinated because I thought this fits. One thing that you probably know, but people have discovered that the issue with world hunger is not a food shortage, but the logistics behind food distribution. So there are different ways of thinking about this. We need to improve access to food by looking at production strategies, trade agreements, and food aid, new technologies. This is sort of where we'd like to be able to think a little bit more flexibly. And then finally, another version of this, we know that a peaceful world cannot long exist 1/3 rich and 2/3 hungry because of this association between undernourishment and conflict. That here's equality, equity, and here's the current reality, unfortunately. And this was a comment by Jimmy Carter that I thought was very appropriate for us thinking about these problems. Okay? Thank you very much.